All right, welcome everybody. I'm glad to see so many participants joining us. Hopefully this will be helpful to you as we go through the practicum, what it is and how to go about it. Just a quick disclaimer before we start. This meeting is being recorded, so if you have privacy concerns of any kind, you can leave your video off at all times. So just a heads up, it's being recorded and it will be on our website. So if you want to refer back to it at any point, you can go on our website and rewatch it as many times as you'd like. Just a quick introduction. My name is Bianca. I work in the advising office for global disease biology, along with a couple of my colleagues that are also on the call, Francesca and Saranjeet. They're going to be watching the chat, so if you have questions as I'm presenting, feel free to drop them in the chat and they can kind of interrupt me and ask me. And then I might not answer your question right away because I might save it for the panel later on. But please feel free to put questions in the chat as they come up. Alrighty, so this is what today is going to look like. We are going to just briefly go over what the global disease biology major is. Most of you are global disease biology majors or faculty, so you probably already know the program. But just for those that are maybe not from our major or department, I can give a brief overview. Then we're going to talk about the practicum project. That's what the focus of today is. And then briefly mentioning what the mentoring process is like. Then we're going to have actually one of our faculty talk about remote research ideas, which I'm very excited about hearing about. And then lastly, we're going to have a panel of students and faculty members, and we have some alumni with us too. So very excited for that. If you do have questions for students, past students and current students, or faculty members that have done mentoring before, you can save them for the panel or put them in the chat if you think you'll forget. Alrighty, so for starters, what is the global disease biology major? I like to always say that it's one of a kind in the whole world because it's an undergraduate program that's focused in One Health, this concept of One Health, which I'll explain in a little bit. There are two unique things that make this major special compared to other majors on campus. One of them is we have 25 units of restricted electives. So it's a little bit more than the normal amount of electives that majors require. And this allows the students to kind of specialize in a topic that they are interested in or that will help them in their future career. And another portion that will help them super specialize is the practicum project. I apologize for the announcement. All right, I apologize for that announcement. I didn't want to have to talk over it. So very quickly, this concept of One Health is the center of the GDB major and program. And one way that I like to describe it, most of you might be familiar with this, and if you are familiar and you would like to add to my definition of it, please feel free to put it in the chat to enlighten some of the other people that are in the chat in the call as well. The way that I like to define One Health is kind of this interconnection between the health of animals, plants, humans, the environment. They're all connected to each other, they all affect each other. And maybe another way to look at this is to think of it as a way to approach disease from multiple aspects. So it's societal impacts, personal impacts to individual people, the discoveries behind disease and how they progress, how they evolve, diagnosis and treatment of disease, what goes into disease, whether that's studying vectors and how diseases are transmitted that way, or looking at how they progress physiologically in the host. So multiple aspects of One Health. It's a very interdisciplinary approach, which I think should be the future of how we solve problems in the world. So it's a really cool way to look at the world. And one way that students can actually apply this is through the Practicum project. Like I said, the Practicum project is a way for students to apply this approach of One Health and to kind of solidify which area of One Health they want to look at things. So the goal really for the Practicum is to experience the whole scientific process. And usually that's, asking a question or addressing some sort of problem that they recognize, then doing some background research and eventually coming up with some sort of hypothesis. And I know a lot of you that have done research before, especially the faculty that are in our audience, you know that sometimes these three steps are kind of all melded together into one big task at the beginning of a project. But just for the sake of having a flowchart, I just had it in this order. Eventually, when you have all of the background research and hypothesis and what you're looking for, you start the process of data collection. And this is where students learn a lot of skills in how to approach different disease issues and health issues. And then the analysis is also another portion where students can learn very valuable skills in how to approach different issues and disease. And then the final step is the Practicum report, which is the very exciting portion for students, because that means that they have finished an entire research project with the help of their mentors. It's a moment that I think makes all of us proud. The alumni that are on the call can probably tell you that. It's a way for students to look back and say, wow, I really did this by myself, or with the help of other people, but you did it. So it's a really cool way to end, I guess, your undergraduate career with this experience. So here's some logistics of the Practicum project. Usually it's a 1 to 2 quarter project. Sometimes that includes training. If the training is a little shorter, sometimes faculty members or lab members would like to train the undergrads for a little bit longer. So it ends up being a little bit longer than 1 or 2 quarters. We do provide students with research units, so the GDB 189 units for students students get 3 units of GDB 189, and they can do research for 9 hours of the week. So that usually includes collecting data, whether that means doing surveys, doing doing experiments, or conducting interviews, anything that's data. I apologize for that again. And then at the end of their project, once they have collected all the data and done all the analysis, there's the one GDB 189D course, which is 1 unit, giving students 3 hours per week to write up their report, get it all ready for submission, and then eventually submit it for approval from our master advisor, Dr. Laveau, who you'll get to meet during the panel. So those are a little bit of logistics for the project. And here is what the mentor role is for the Practicum. First of all, the title of Practicum mentor is technically reserved for the faculty member that's going to be the instructor of record for those research units, GDB 189. Usually that's the PI of the lab or the head of the team that the undergrad is taking part in. So that will be the person that grades them pass, no pass on those research units. That does not mean that students can't work with other people in the lab or in the team, though. A lot of times students will actually work with, directly with a grad student or a postdoc or any other lab member, and that will be their kind of first contact in the lab and first point of contact for questions and guiding them through the process. But the PI or the head of the lab will be the person grading them, a person with a teaching title. If the undergrad does work with multiple people in the lab, the practicum mentor, so the person that graded them in the research units, gets to sign off on the final project. And if the grad student or postdoc wants to sign off as well, they can also sign off kind of as a sign of approval for the undergrad's final work. So it's a very exciting part, like I said. It's the completion of this whole project that seniors have gone through. So speaking of practicum mentors, Dr. Tiffany Lowpower is a current and previous practicum mentor, and she's going to share with us a few ideas on how to do remote research with undergrads. Hopefully this is enlightening and gives some ideas for some of the faculty that are in our midst today. But students, you should also pay attention because these could potentially give you ideas on things to suggest to potential practicum mentors. If they don't really know how to navigate the remote environment, you can always suggest something to them. So Dr. Lowpower, I will let you share your screen and introduce yourself.
Yeah, so I've unmuted myself, but I don't have the permissions to share my screen. So you might need to make me a co-host in the participants window. There we go. Cool. So now, oh, yes, I'm gonna stop sharing yours and pop this one up. Okay. So these are just a handful of projects that I've kind of thought about in terms of thinking about how to transform my lab's research into work that anyone can do remotely. Because my grad students and postdocs are also working staggered shifts in lab and at home. So this project is a literature meta-analysis. And this is a project that Kyle Chipman, who graduated from GDB in spring 2020, participated in the spring. And it was our response to COVID and trying to figure out how to help him get a practicum project since he couldn't come to lab and none of us were in lab. So this is a literature analysis, and because I'm an expert in this one group of bacteria, Ralstonia, that cause wilt disease, I was familiar that our Ralstonia community has published hundreds of papers that use a standardized method to describe the taxonomy and diversity of Ralstonia. And I knew from having read some of these papers that a gap in my own knowledge is really understanding how these diverse groups of this pathogen are spread around the world and where they are and what hosts are, what plants are catch a lot of disease from Ralstonia in different locations. And I knew that this information was fragmented in the literature and no one has, brought it all together in a systematic review. So Kyle and I decided to use a Google Scholar-based search to create a large database of all of the strains of Ralstonia that have been classified by this schema. And yeah, so since this is a particularly large schema, there's hundreds of papers. Kyle entered data from about 35 papers, and so there was still more to contribute. So I recruited two new students, Maria and Jason, for this fall. And so this is just, kind of showing that there's four major groups of this bug. And but if we look at some of the smaller clades, they have more localized distributions. but we don't know the big picture across the globe and across all of Ralstonia. So if something like this project sounds interesting to you, there's a little bit more information about how we actually set it up on this web link. And I can have Bianca send this out to the relevant lists. And so, yeah, and so, you know, that project, it works for Ralstonia, but I think it could work for any pathogen group. If there's a research mentor who's familiar with a common way that the organisms are described, you might be able to find all those papers and do a meta-analysis on them. A second project is an approach to look at bacterial gene functions. You could also look at fungal gene functions, but I'm a bacteriologist. The goal of this project is to characterize the functional diversity of groups of microbes. So if you look at this kind of figure, you see a lot of different bacterial strains, and there's a phylogenetic tree on the left. And then what the table to the right indicates here are whether certain genes are found in the genome of each of those bacteria. And so this is a trait that allowed bacteria to consume certain carbon sources. And there's a lot of genes in the pathway. And what we found previously are that most strains of this bacteria have all of these genes, but there have been a few genetic losses in different groups. So characterizing that genetic diversity allowed us to then take some of the functional assays we were doing in the lab and say that this was generally a broad trait in the group and not just a trait of our specific model strain. And so if there's a lab that you're interested in, or your lab is focused a lot on molecular biology and gene functions, there might be some favorite genes you have and groups of microbes that either are taxonomically related, evolutionary related, or ecologically related and share a similar niche. Looking for the distribution of those gene functions in the genomes of the organisms could be a really useful exploratory discovery based scientific question. And so my group, a PhD student in my lab, Stratton, mentored two students over the summer, a GDB student, Elva, and another remote student, to characterize distribution of all of these genes in several bacterial genomes. And so it works because we can use some web-based tools. There's a lot of genome databases. And then, you know, the mentors have some specific knowledge of which genes are interesting to look at. And then this paper is published if someone wants to see how it led to increased significance of a study. Another approach that would be useful, research labs sometimes generate more data than they have time to analyze, but they might have very reproducible, you know, a good idea of ways to analyze that data. For groups that produce a lot of microscopy images, sometimes there's ways to quantify that microscopy into, you know, more detailed information than just qualitative images that are put into papers. And so you might be able to get a large data set shared with you via a box.com folder, and then have your mentors train you in how to look at these images and then classify it by different groupings. So here we're looking at plant stems, and within the vessels, the xylem vessels, which transport water in plants, some of them are clogged by bacteria, which are stained this ever so subtly darker blue. So if you look closely at the microscopy, you look for little cyan vessels and then for amounts of bacteria filling them. And so with this kind of quantitative analysis, we're able to see that over time, the bacteria start to clog up the vessels. But there might be other sorts of imaging data sets that labs have that they can bring you in to help analyze those data sets. or there might be genomics data sets. Especially with omics these days, there's a lot of ways to create more data than we have time to necessarily parse. And then a fourth idea is it wouldn't necessarily, and maybe it could lead to doing a bioinformatic analysis. And this one could actually be complementary with any of the other projects. But there's this really nice online class through the iBiology website. It's a self-paced online class called Let's Experiment, a guide for scientists working at the bench. And I have a new research lab that we started in July of last year. And this is actually a online class that my lab was working through in January and February before the pandemic hit. So we didn't realize we'd be doing so much online learning. We were trendsetters. But this course is really nice because it has a lot of case studies and short videos that really explain how to design robustness into experiments. So thinking about, for the biological question you're asking, what is the control? How much replication do we need? And other factors that help you design an experiment that actually, tests a principle of biology. So I think that this is a kind of project that would work really well for an undergraduate mentee who is paired with someone in the lab, probably a newer grad student or a newer postdoc in the lab, who's at the cusp of really developing their own project as well, and is probably trying to plan out their path while they're doing the remote part of their job this year. And they might be able to, it might be really beneficial for them to actually have an undergraduate mentee who's an apprentice who helps, helps them talk through some of what they're thinking about. And in the process of them, working together with the undergraduate, both will potentially think about the project in a deeper way. So this is it's available online. And then the tools from it, I think, will really help the grad students and postdocs continue to prioritize their time well and design good experiments. There's a lot of nice tools like this flow chart of deciding, which experiment do I do next? But there's also a lot of worksheets in terms of just, having you step through a research question in a really nicely organized way. So I'm going to send all of these slides to Bianca, and she can forward them to students. And I think that, when GDB students are contacting professors to ask about research opportunities, I encourage you to, look, if there's certain types of these questions that you would like to have more, feel free to use this slide and ask the professor, is there a project like this suitable in your lab and someone who would be able to mentor me on it? And yeah, so that's all I prepared.
Thank you so much, Tiffany. That was very helpful. Yeah, it's always good for undergrads to reach out with some ideas, I think. It at least gives them, something to work with. And I think another thing to point out is this can be used for a number of things. Dr. Lowpower is a plant pathologist, but this can be used in a number of different categories of labs. So if plant pathology isn't necessarily your thing, you can still use some of these ideas for suggesting to other lab members or other PIs. So thank you, Tiffany. That was very, very helpful. We're going to introduce some of our other panelists. Just a reminder again, put your questions in the chat and we can go through them and answer them with our faculty and our students and alumni. So let me just do a brief introduction. You've already met Dr. Lowpower. I will let you all introduce yourselves. So Dr. Laveau, if you would like to introduce yourself.
Sure thing. Can you hear me all right?
Yep.
All right. So hi, everyone. Thanks for coming to this event. Thank you for Bianca for putting it together and all others at the office and explaining about the practicum. If you don't know me, I'm a professor of plant pathology at UC Davis. I'm also the master advisor for the major, Global Disease Biology. I also teach several courses, SAS 13, Disease and Society, GDB 103, the microbiome of people, animals, and plants. And most importantly, I suppose, for this particular event is the fact that I am the instructor of record for GDB 189D. So that's the part of your practicum where you write up your experience in a mini manuscript. So we can talk more about that if you have any questions later on. Thanks.
Thank you. Dr. Lopar, do you want to introduce yourself in a more formal way?
Yeah, I guess that would have been a good way to start the presentation as well. So I'm an assistant professor in plant pathology, and we have been in research lab since July 2019. So half of my time being a professor at UC Davis has been during the pandemic. Yeah, so it's been exciting to pivot to the pandemic lifestyle. But I feel like I was already like running and having to jump to different directions as we hit different hurdles. So this is just the latest one in front of us.
As always, there are hurdles in science. Dr. Neil McRoberts, you can introduce yourself.
Yeah, hi, good morning, afternoon, everybody. I'm Neil McRoberts. I'm like Johan and Tiffany, I'm a professor in plant pathology. Some of you I'm teaching at the moment. I teach the epidemiology GDB 101 class to the GDB major. And I've had several undergrad interns working with me, either as part of the GDB practicum requirement or doing research internships with me for several years. So plus in quite a few of the last few years, I've picked up a few stragglers, people who found it difficult maybe to find a lab to work in. So I've mentored a lot of different subjects through the GDB practicum exercise. So if I can help share experience of what that's been like from my perspective and what leads to success and failure. Actually, I don't think we've had any failures. So what leads to success, then I'm only too happy to answer questions.
Yes, there have been many, many GDB students that have worked with Dr. Neil McRoberts, and he does very interesting research, so yes. Our other panelists are alumni and current students. So we have 3 alumni with us, Nicole, Amanda, and Emily. And 2 of our 3 peer advisors are going to be our current student speakers or panelists, Indira and Hannah. I guess I will let you all introduce yourselves as well. So in order, you can tell us what your project was or is on, and yeah, what year you are if you are a current student. So Nicole, you can start.
Hi, I'm Nicole, and I graduated as a GDB major just this past spring, so in June. My practicum project was with the Food and Nutrition Department at Davis, and it was on food insecurity prevalence of United States college students. So it was like a modified meta-analysis, so kind of like pulling from different literature and coming to a hypothesis that my, how do I call it, my mentor and I came up with.
Thank you. Amanda?
Hi y'all, I'm Amanda. I just graduated this past September, and my practicum project was on studying plaque psoriasis using a mouse model and figuring out, investigating these new therapeutic targets for treating plaque psoriasis. And so I'm continuing to study chronic itch and pain in the lab that I did my research practicum in, and I'm currently interviewing for MD PhD programs. So happy to talk to students about the practicum or that.
Very cool. Emily?
Hi, Hi everyone, I'm Emily. I am an alumni. I also graduated this last spring. And my practicum project was about characterizing the prevalence and virulence of an avian coronavirus. And I'm currently studying for the MCAT right now. I plan on applying to medical school. So I just want to let you guys know that you can still do research in different fields, animal health. You can do plant pathology and still have an interest in human health or want to go to medical school and vice versa.
I love you pointing that out, Emily. That's what One Health is about, right? Studying disease from multiple different angles. So, Indira or Hannah, you can go next.
I'll go. I'm next in the order, I guess. My name's Indira. I'm one of the peer advisors in the GDB office. I'm in second year, so I'm not quite at the practicum stage yet, but I'm doing research with the Social Environment and Stress Lab over at the Center for Mind and Brain, and I'm also doing research on COVID in pregnant women and kind of doing data mapping for our region with the Perinatal Origins of Disparity Center. So yeah, my interests are kind of in public health.
Very, very cool. You have plenty to choose to make a practicum project out of that. Hannah?
Hi everyone, I'm Hannah. I'm a fourth year GDB student, and my practicum project is studying how iron intake affects the declarative memory development in infants. So it's been really fun, well before COVID, to like go see the babies and do the little memory assessments with them. So I ultimately want to go into public health in the women, mother, child sphere. So yeah.
Very cool. I'm also fascinated by that field in public health. I'm going to unshare my screen so we can all see each other, and then we can start just doing some questions. I see Dr. Lovo already answered one of the questions on there. So I will do this question for our faculty members on the panel. What is the structure of your lab or team, and where do undergrads fit in that structure?
I'll go ahead and lead. So for my lab, we have, we're a newer lab. We have 2 graduate students, one student who's completing a master's and one student who's completing a PhD. They joined a year and 6 months ago. And we also have a postdoctoral researcher. So postdocs are scientists who have obtained their doctorates, and they've graduated with a PhD. And they're continuing on to, it's kind of like medical residency, but for academic research, I think. I don't actually know the medical side as much. And so that's the main structure of my lab. And then undergraduates fit in at multiple levels. So depending on whether there's someone in the lab who I think has a project that, is amenable to carving out and mentoring an undergraduate on. So my PhD student Stratton is has mentored 2 different students because his project makes a lot of sense for that. And he also was the first to join my lab. And then for the Ralstonia Diversity Project, that doesn't tie into any specific project that the grad students or postdocs are working on in my lab, but it's something that I know a lot about. So with those students, I'm working directly with them.
I can add some to that. So that what Tiffany just described is not an untypical way that a lab would be structured. You have the PI, you have postdocs, you have graduate students, and undergraduate students really become a member of that lab once they join the lab in such a way that they join lab meetings, they see the dynamics of the lab. Obviously, things are a little different now during this time of remote lab activity. But for example, my lab does meet over Zoom, and we have good discussions, and we keep the train going. We keep the research going. And so it gives undergraduate students really an opportunity to experience what life is in a laboratory. And I always invite them at the end of their stay in the lab to give a short presentation, informal, just for the lab members, to share their experience. And very often that is it's a positive experience. But we've also had students who said, boy, I really realized that research really isn't for me. And that'd be a good time to find that out. That is something you have to experience before you can write it off as something you don't want to do, or something you realize that is very exciting to do and that lies in your future. So that's how I see the way that undergraduates get incorporated in the lab. It's exciting. I think should be exciting for everyone.
Yeah, I think presenting is one of the ways that you can kind of solidify what you've learned through the experience. So I always like that. Neil, do you have anything to add? I know your team might be a little bit different.
We're different, but not so different. So that we one thing that people should know is that we don't have a wet lab. So I don't have any lab space as such. My group works on data collected in the field, and we work on questionnaires, and we work on large data sets collected by state and federal regulatory agencies. And so a lot of what goes on in my team is data analysis and interaction with stakeholders. And but with that slightly strange difference aside, how the undergrad interns fit into the lab isn't that different from what Johan and Tiffany have described. There's a couple of big programs that are run by staff in my group, and so there are opportunities for people to fit into those bigger programs, and then the staff member who project manages those big projects would probably be the main point of contact, at least on a day-to-day basis. But one thing that I do try and do is, so even before the shutdown, a lot of the work that my team does, because of the nature of it, we work remotely a lot of the time anyway. So we use Slack a lot for interaction and for keeping in touch. So anybody who joins the group is invited to join the Slack, and then there are sort of separate channels specifically for the interns to speak to each other and to develop their projects. And I try and have a at least monthly, if not more frequent, meeting just with me and the interns to touch base and see how all the different projects are going. Some people work directly just with me in one-to-one projects. Some people fit in to those bigger projects, and occasionally we get the sort of special teamwork projects where we need, say, a data set to be crunched pretty quickly, and so everybody is like all hands to the pump, and all the interns help with that, and they divide up the work between them, and they work on in an intensive way on that until it's done, and then come back to me. So there's a lot of different ways of working. And then in addition to all of that, there are students who just work with me on their own projects. They come with a practicum project already in mind, and all I do is basically provide a sounding board and a critical reviewer, and I act as a reader and editor, and I help them just by being a sort of scientist and having an opinion about what they're doing. And there's been projects on everything from microplastics in the environment to hormone levels in livestock. So those students aren't so much really part of the lab, but they still are invited to participate and take part, and they either do or don't. It's up to them.
Thank you. Maybe a follow-up question to this first question was from Gamar. How long does it typically take to train undergrads in your team, and I know Tiffany just answered shortly. Is there a different answer from Johan or Neil?
It depends on what the outcome is going to be. To do some simple things, it doesn't take long at all, and you may already have the skills you need to do that. If there's specific tasks that need to be done, it may take a month or so for somebody to learn how to get comfortable with, say, learning a particular piece of R code to be able to run a type of analysis and that kind of thing. So but it varies a lot. It can be no time at all to several months.
Yeah, so what Neil said is what I would say as well. It really depends on the project that the student will be working on. One thing to really realize is that a willingness to learn is going to be a big factor in this as well. So, take an active stand on if you are being trained to absorb it and to take that training seriously. It's a big investment for people that work in a lab, whether it be a PhD student, a postdoc. I highly encourage you, if you can, get an experience in a lab that's maybe before you start your practicum experience is a good idea, simply because you get to learn who the folks are in that lab, what kind of research they do, where everything is, and that will give you really a head start on then developing that into a GDB practicum experience. So if you have that opportunity, I would definitely take advantage of it.
And to follow up on what Johan said about getting in, starting early. So I think especially for, freshmen, sophomores, juniors, I think it's going to be harder for labs to be able to accommodate undergrads period into labs, because we have space restrictions in order to keep all the people density low so that we don't spread the pandemic. But it's like for all students, something that I think is a potential, a creative positive about the pandemic and remote experience is you might be able to ask some professors when you're emailing them, do they have a lab meeting that they would be willing to let you join? A lot of us are doing these lab meetings over Zoom. And it's actually, so there's no, necessarily room restrictions. We don't want to open them up necessarily for 50 people, because then it's, hard to manage a group and have, good discussions. But it could be a way for you to, maybe do a month in a few different labs and just get a feel for the types of questions that those labs are discussing, the types of approaches that they're using, and whether, and if there's something that really excites you, follow up. If there's not time in the Zoom, send an email being like, that is so cool. Is there a part of that I can read up on more and think about developing into a practicum project, even if it is a bioinformatic or a literature-based project. Reading the literature is a super valuable skill, and reading it helps us develop good ideas. So, it's really fun to move liquids in lab, but definitely the reading and bioinformatics approaches are really valuable too.
Thank you all. I do want to point out Dr. Laura Kerr. Thank you for putting that in the chat. I was going to point out that, if you're doing clinical research or working with a clinician, sometimes it'll look kind of differently. You sometimes will be working directly with the clinician. Sometimes you'll be working with a research coordinator. Sometimes my research project when I was a GDB student was with a clinician, but I also collaborated with a wet lab on campus. So it might look very different, the structure, but I mean, essentially, the idea is you're working with that mentor probably a little bit more closely since they're heading the project. Thank you for pointing that out.
I'll put a plug in for over on the Sacramento campus. You know, we're even less far apart than we are usually because the causeway doesn't matter with Zoom, right? But not that it usually matters anyway. But you know, there's lots of people over here who do work with data sets, for instance, like CDC data sets or large national databases that focus often on human health. But you know, there's a lot of things that can be done that way. I also do qualitative research where you interview people, and that can be done just as easily through Zoom as in hospital rooms. So there's definitely still opportunities over here.
So there are some other questions here that I think might be more appropriate for our students to answer, or our students are more than able to answer. And I want to be very mindful of the faculty panelists' time because I know you are busy people. So if you have to go at any point, thank you so much for participating and answering all these questions up front. If you want to stick around, that's also fine, and answer more questions. So thank you. I think maybe we'll take a few minutes, like a 2-minute stretch break, and then we'll come back and answer some more questions for more specifically to the students and alumni. So go ahead and stretch a little bit. All right, everybody. If the panelists are there, I'm going to start asking you some questions. What are some of the best ways to reach out to possible mentors for a practicum project? So maybe the students can share how you reached out and how you got your practicums.
I can start. So I was part of a couple of labs before, and when I was reaching out to those labs, there are programs on campus that cater to getting undergrads involved in research. So I was part of the it was Research Scholars in Insect Biology. And I initially started there because I was interested in vectors and disease. But you can, so I would say look to programs that are on campus, and you can find those through like your email. But also if you go online to different departments, they have like a staff list. And so you can click on the staff list and look through all of the staff that are part of that department. And if they have any research, it'll also have like their associated research on it, and usually their email. And so I found my Practicum mentor by doing this, and I had emailed somebody and they said that they actually had a colleague that needed help. So it's just a matter of taking the time to send a lot of emails, and usually the researchers are very nice and they'll either point you in the right direction, like, oh, try contacting this person, or they'll gently let you down. But I would say you just have to start sending out emails, and you can start by just looking at different staff lists. Okay, someone else go.
Yeah, I can follow up on that. I found my research practicum, or the lab that would be part of my research practicum, on Aggie Job Link. I think that's outdated now and it's called Handshake. But I found that in my first year, so I really stress being open-minded to different avenues of research or finding research, but also different labs. I was interested in neuroscience, so I looked in the neurophysiology behavior department and found a lab that I think I would never have considered before, but had an opening, and I was just interested in learning the techniques. But then it became something that I was really passionate about, and really influenced my career choice. So definitely being open-minded, but balancing that with like reaching out to people that you're genuinely interested in, because like our faculty members have said, it takes a lot of time to mentor a student. So though you're reaching out to a large group of people, make sure that you know something about what they're doing, and that you're genuinely interested in putting into work for your work for your practicum project.
I think the question was like, how did you find your practicum project and your mentor? So there are a few resources that I used, kind of all the methods that they just mentioned as well. Handshake is one good place to reach out to people. The GDB newsletters are pretty helpful as well. They usually have send that out at the end of the newsletters or something of the sort. Referrals are good too. So if you have a friend, like even maybe some of the alumni are here now, like maybe they know of faculty members that are looking for somebody to replace them. So that might be good too. I know it's a little bit different now with the pandemic, but I also reached out to my jobs on campus, and I know that a colleague of mine ended up doing her GDB practicum project. She's working on it now, like while working as well. So that's an option too. And even if your on-campus job isn't maybe like back, like you're not back on campus right now, maybe they have other research opportunities for you as well. And I think that was it.
I do have a follow-up question to Amanda. Do you recommend emailing multiple faculty all at once, or emailing one at a time and kind of waiting for a response?
Yeah, I think it's completely fine to email multiple faculty. And I think from talking to friends, I did mine through Aggie Job Link or Handshake. So there was like an application process, but through talking to friends who did reach out purely through email, I think just making sure that you cater your email to their genuine research interests, and think about some ways already that you could be doing your research and ideas for their project, or just being genuinely interested and saying, hey, are there papers I could read, are a way to get some good responses. You don't want to just do like a cold email to like 50 people of just, I need someone to mentor me, anything works, so.
Yeah, I think the theme here is genuine interest. Show a genuine interest in what they're studying. You don't want to waste anybody's time, your time, faculty's time. So, a topic that you're genuinely interested in is usually going to be a better fit for both of you. Let's see. Maybe this is a question that students or faculty can answer. If we have no experience in a lab, are we still welcome to join a lab? And what, like, is that any different from someone that has already experienced before?
I can say something on that. So the first lab that I joined, I was a freshman, so I had zero research experience. But if I have learned anything about my time or doing research at Davis, it really is kind of along the theme. If you show that you're hardworking and you want to learn, even if you have no experience, your mentors are going to see that you are a hardworking person, you made an effort to come talk to them. If you do some research on your own about what they're doing in different ways that you can educate yourself, that's really going to help. And then the lab that I actually did my practicum in, I took no vet classes. I had no experience in histology, so I didn't not know how to look at a slide and tell the difference between a macrophage and like a neutrophil. And I was just like, I don't know what I'm looking at. I don't know what a trachea looks like under a slide. And I had to know that kind of stuff. But my PI and the other staff that I worked with, they like teaching. They like teaching the stuff and sharing their research. And so they're going to take some time to help you if they know that you're going to be willing to learn and put some time in. And so I took the time to like watch YouTube videos on like pathology, and I had to like look through books in a library on like what the different tissues look like. And so it takes some work. It goes both ways.
Yeah, I second that. I also had no research experience, and I think that that highlights the importance of mentorship. I think when you take time to explore different labs and you get to know people and meet people, you should definitely talk to the graduate students who probably will have, or the postdocs or whoever has the most time to mentor you, and see if there's a relationship there that you could foster and learn from, because you will be learning on the job most likely. And so I think that scoping that aspect out of a lab, not just the research topic, but like, do folks have the capacity to mentor you in the way that you need to be mentored, is really important.
I don't know if the faculty maybe have anything to add.
Yeah, well, just kind of picking up on that last point that was made, I think it's really important for you to, it's not all about the faculty interviewing you necessarily to find out if they want you. You've got to find out exactly like Amanda said, do they have time to help you? Or are you going to sign yourself up to go and work in a lab where the PI is so busy that you never see him or her, and they don't, and basically you don't learn anything. You're just kind of sitting there not getting anything out of it, and that's the last kind of situation you want to be in. So don't be too don't forget to think about yourself, is what I'm trying to say, because you need to get out of it something useful. And so that's an important thing to remember. But just also, I'll just say that the comments that have been made are spot on. There are, several different reasons why we might take on people for practicum work. And one of them is just that kind of general recognition that the next generation of people need to be trained, and so we have to kind of make sure that that happens. And so there's sort of, it doesn't matter if you don't have any experience coming in, we understand that we're going to train you. That's what the whole point of the practicum is. So there's that kind of opportunity. And then there are more focused opportunities where there's a project that's already ongoing, and the PI will be looking for somebody who's maybe got some lab experience already to help out, support a graduate student or a postdoc who needs a relatively experienced pair of hands to help them. So just be aware that both kinds of opportunity exist. And so you might not be, if you don't have any experience at all, you might not be suitable for all of the opportunities that are out there, but you're suitable for a subset of those opportunities.
And I want to chime in there with it's hard because it's about your own psychology and how you feel about things. But there's no logical reason for you to be afraid and intimidated. You know, we all were in your same spot at some point, not knowing exactly what we were doing. And the key thing that like makes someone be successful in a lab versus someone who just, you know, clocks out at the end of the day, is whether you get engaged, like the senior students here have talked about. Do you try to ask questions? And then, you know, you talk with your mentor some, and then, you know, definitely going beyond the conversation and trying to read up on it to reinforce what they said can really help. But even if it's as simple as, you know, you get shown how to do something, you do it on Tuesday, and then you show up on Thursday and they're like, okay, do it again. It's okay to say, I don't remember. You know, we don't expect you to be like, you know, a perfectly programmed robot immediately, or even like after a while, after a break. So, you know, it can be very, it's hard socially sometimes to admit that you don't know what's going on. But you know, if we're training someone new and they ask no questions, we know that they just aren't asking the questions. And it's like, yeah, like, I don't know. We really appreciate the students who ask questions.
The postdoc, oh, sorry, go ahead, Neil.
Well, I was just going to say, can I share a screen and just give you one kind of case study of like one recent intern's history to kind of give an idea of what's possible. I think because people relate well to examples, right?
Yes, go for it. I just made you a
Okay.
co-host, so you should be able to share.
Okay. Okay, so hopefully you're all looking at my web browser now, right? Okay, so this is the homepage of a journal called Entropy, which is all about uncertainty and information. And a colleague of mine is just wrapping up being the guest editor for a special issue of that journal about applications of information theory to epidemiology, okay? And so I'm going to scroll down here. So here are some of the papers that have been published here. And this very first paper on the list is a paper that I published with the guy who's the editor. And the name in the middle there, Jennifer Kopetsky. Jennifer was a biochem major. She did the GDB minor last year. And she took GDB 101 with me in fall quarter of 2019, and she asked after class if she could do some extra study intern with me, and she worked on various data analysis projects, just fairly simple data analysis approaches. And when she graduated in June, she kept in touch. She stayed engaged with the group via Slack. She, after graduation, went to live in Canada. But as we were writing this paper, Gareth and I were writing this paper, we came across a problem where we found an old paper that we'd both missed in the past, and we needed to check to see how much that paper had been cited in the literature, and basically make sure that we didn't miss a trick when it came to the work that we were doing. And so Gareth asked if there was anybody around who could help do that work. And I reached out to Jennifer and said, hey, could you do a systematic literature review for this publication? And she did the work on that. And she didn't really understand all of the technical details about what the work was about, but she did a fantastic job of making sure that we didn't miss anything. And she contributed in the end to a significant extent to the paper we were writing. And so she ended up as a co-author on that paper that just came out a few weeks ago. And probably it's not going to be significant in her future. I'm not quite sure what she's planning to do at the moment, but just to point out that started from her staying behind after class to ask if she could work in the group. she did a whole load of stuff that wasn't connected with this, and then she ended up doing this nice piece of work that ended up being included in a paper that's just come out. And that's, you know, and that's a possible trajectory for anybody, to be honest. And Jennifer's key attributes that really made her a standout intern for me, she was hardworking, she was meticulous, she was really engaged in every topic that we worked on together. She made sure she really understood it. She got really fascinated in everything that we did across a wide range of subjects. And she, when she was given something to do, she did it on time and usually went further than she was asked to go. So that's kind of the sort of recipe for success, I think.
Thank you for sharing that. Yes, examples are always good. And yeah, I took GDB 101 with Jennifer, and she was always really engaged in the class in general, in our discussions in class. So if you take a class that you're super interested in, it's worth talking to the professor and just, you know, discussing your interests and seeing if you can join whatever project they're doing. So I want to leave some time so that we can look at examples of projects. So I'm going to answer these last two questions very quickly. The first one is, do you develop your question for the practicum before searching for a possible mentor, or is that something you develop with your mentor? It can be both ways. Dr. McRoberts mentioned earlier that sometimes students will come to him with a project that they have in mind, and he will just kind of be their mentor through the scientific process, right? Being a critical eye and looking at it and seeing what are the possible roads to go, what are the possible things to improve on it. So sometimes students do have their own ideas and they bring it to a mentor or to a professor just to kind of help them in that aspect. But most of the time, I would say students go into a lab because they're generally interested in that topic. And then they will come up with a project with either the PI or the grad student or someone in the lab that they're working with. So it can go both ways. And GDB students have done both for their practicum projects before. And then the last question is, would training be something that's included in GDB 189 units, or is training something that would be done prior to signing up for GDB 189? I think this would kind of depend on how long your training is. If your training is going to be a whole quarter, I would say if you want research units, maybe ask your lab, your PI or your professor if you can do research units, like internship units, you know, like PLP 199 or something like that. So you can get units but get trained before you actually start your practicum project. If the training is going to be, you know, a week or two out of your whole practicum experience, which I think that's too short, but if it were the case, you can take 189 and train within that time frame. So it kind of depends on how long training would take. Are there any last minute questions that anyone wants to bring up? If not, thank you for all the panelists for being here. I hope that this was really helpful in giving perspective from both the faculty side and the student side. I really, really appreciate the faculty perspective on this. I think it makes us a little bit less afraid to reach out to you and ask you questions and ask to join your lab. Like Tiffany put in the chat, I think imposter syndrome is very, very common in our scientific community as we're training. So it's nice to know that, you know, we can reach out to professors without having to feel like we're always on top of it. Yeah, so thank you so much for the panelists. I think the student panelists are sticking around for a showcase of previous practicum projects. If the faculty want to look at those too, you're welcome to come in, but I will start breakout rooms in just a minute.